Hello, biology students. We are now ready to conclude our chapter on RNA and protein synthesis. We're going to finish up this chapter by looking at gene regulation and expression. And so basically, how genes are either turned on or turned off. And so we're going to look at a few things today. We're going to look to see first how prokaryotic genes are regulated. We're going to use a classic example, and we'll talk about that in just a minute. We'll also see then how genes are regulated in eukaryotic cells. And so it's very similar to that of prokaryotes, except of course it's a little bit more complicated, but not the same basic idea. We also are then going to finish up by looking at what controls the development of cells and tissues in multicellular organisms. And so we do have some definitions, but we'll go through what each of these are today. And so let us go ahead and start then by looking at how prokaryotic genes are regulated. We have a group of genes associated together, and because they operate together, we're going to refer to this as an operon. And so operon is a group of genes that are regulated together. A good example of this is what's referred to as the lac operon. Now, lac is an abbreviation for lactose, and so notice we have the three letters os, and so as we talked about before, we know that this must be a type of sugar, and yet it is also the sugar that we find in milk. So lactose, we need to talk about how bacteria also, just like humans, just like everything, needs food, and so it gets it in the form of sugar, which in this case is lactose. And so in order for the lactose to be taken across the cell membrane, it must first be broken down into its two component sugars. So lactose, again, is referred to as a disaccharide, and so if you remember, the prefix di refers to the number 2. And so lactose is made up of two different sugars. It's made up of glucose, and it's also made up of galactose. And so in order for the lactose to get transported into the bacterial cell membrane, it first has to get broken down into lactose and galactose and into glucose. And so we have a picture down here that shows the entire lac operon. And so I want to point out that there are 3 genes involved with this process. We have the LAC-Z gene, the LAC-Y gene, and the LAC-A gene. And so all 3 of these genes together are what's going to regulate the transport of the broken down lactose into the bacteria. And so also notice that we have an RNA polymerase. And so remember, whenever we have something that ends in ase, we know that this is an enzyme. And so we're going to see that this is going to be, again, involved in the transcription of DNA into messenger or mRNA. We have also a couple other points of interest when we're looking at gene regulation. We have what are called operators, and we have what are known as repressors. The region on the operon that is in control of turning on and off genes is called the operator. And so the operator is also known as the O region. And so not named after Mrs. Winfrey, unfortunately. All right. We also have lac genes are turned off by repressors. And so the repressors are then turned on by the presence of lactose. And so the reason for this, as we've talked about before, doing this process obviously requires energy. If there's no lactose present, then there's no reason for the genes to be transcribed. There's no purpose, because that's just a waste of energy. And so the cell is smart enough to realize that, and therefore will block the RNA polymerase from doing its job. So if we look down here at the picture, if, for example, we have the repressor, and so the repressor is present in the case where there's no lactose, and so notice that the repressor binds directly into the operator region, or the O region, and so it prevents the RNA polymerase from attaching to the promoter. So it actually acts as a physical barrier in order to keep the RNA polymerase from doing its job, which is transcribing the DNA to RNA. So notice this is in the case when there is no lactose. And so therefore the repressor can bind to the operator, and therefore these genes will not be transcribed. We're now going to look at a little bit higher level. So we're going to be looking at eukaryotic gene expression. Fortunately, the ideas that we just talked about for prokaryotic gene regulation can be applied fairly easily to the eukaryotes. And so most eukaryotic genes are controlled individually and will also have regulatory sequences that are much more complex than those of the lac operon. And so the lac operon just provides us with a good basic example about how genes are either turned on or off. As we discussed before, there are promoter regions where the DNA or RNA polymerase attaches to and gets it ready to do its job. So these are referred to as Tata boxes. These are regions of DNA that are rich in thymine and adenine. And so there are repeats of both thymine and adenine, so these are referred to as Tata boxes. And so this will help position the RNA polymerase just before the point which transcription begins. And so remember, we refer to that as initiation. And so after initiation comes elongation. Once it hits a stop sequence, then it will terminate, and then it'll end. And so that is then how eukaryotic genes are regulated in a very similar manner as it would be in prokaryotes. I want to talk to you now about a very new technology, so a technology that has been developed in the past 30 or 40 years or so, and it is referred to as RNA interference. We know now that cells contain lots of small RNA molecules that don't belong to those three classes of RNA that we talked about last week. So they're neither mRNA, they're not tRNA, and they're not even rRNA. So they have nothing to do with any of these types of RNA. So why are they there? These small RNA molecules actually play a powerful role in regulating gene expression. When the original scientists went back to sequence the human genome, they were surprised to find out that the number of genes that actually produce proteins is relatively small. A lot of the genes in our DNA is in relation to this regulation. And so this regulation is important because it allows for the turning on and off of genes. There's a specific enzyme known commonly as dicer, and so it will dice small stranded RNA, dsRNA, into small interfering RNA, siRNA. Once this siRNA is made, it will then form a silencing complex which will destroy any RNA sequence that is complementary to it, which will lead to mRNA degradation. And so, much like the repressor that we talked about when we're looking at gene regulation, this actually takes it one step further. And so, if the siRNA finds a complementary sequence of RNA, it will actually then target it for the cell to then break down that mRNA. And so, by blocking gene expression, we can use the siRNA silencing complex, and it's known as the RNA interference. And so the diagram on the right here shows this process. This is a very important tool for molecular biologists and other people. It allows them to basically determine the role of different enzymes within the body. So if they're looking for a particular role, they can use this RNA interference to basically degrade the mRNA and therefore see what effect that has on the cell. So this has proved to be a very valuable research tool that has been developed in the past 40 years or so. We're going to end our topic looking at development and differentiation within an organism. Regulation of gene expression is important in shaping the way an organism forms. And so think about the fact that you only have one head, you have a couple pairs of arms, you have a couple pair of legs. You obviously don't want to have an arm where your head should be or a leg where your arm should be or vice versa. There are specific genes during development that are turned on or off that will lead the cell to develop the correct body parts in the correct positions or locations. And so differentiation is the process in which cells become specialized in structure and in function. And so there are different kinds of genes that are involved with this. We have a set of master genes that regulates organ development. These are known as homeotic genes. And so think about, again, the process of homeostasis, and so it comes from the same roots. We have homeobox genes, which are also known as Hox genes. These will code for specific transcription factors that will activate other genes important for cell development and differentiation. And so, as we talked about before, one of the favorite models that geneticists like to use is the fruit fly. And so the fruit fly has relatively few amount of genes, and they are able to be grown up made very quickly, and they're able to look at the effects very quickly. If, for example, you were to mutate one of the Hox genes on the fruit fly's head, instead of getting the normal antenna, you would actually, this is a little freaky, you would actually end up getting a leg coming out of the head of a fruit fly. So imagine if you're this poor fruit fly, you're supposed to have an antenna, but instead you have a leg growing out of your head. I know that would freak me out if that happened to me. The leg obviously does not have any use, but is not supposed to be there. And so by changing the Hox gene, we see that that is going to cause a developmental problem with that particular fruit fly. And so the timing and the regulation of genes, as we can see, is very important for the regulation and the development of an organism. And so with that, we are now done with chapter 13. And so we will review in a couple weeks then over the chapters, and so then our test will be in a couple weeks. And so I hope you guys learned a lot, and I will talk with you guys again soon.